Hello everyone, my name is Marie. I am from MaxWell Biosystems and I will be your host for today's webinar. And yes, so we are starting a new series this year, and this is the first one that we have for this webinar for 2023. And first, let me introduce to you our company. So we are MaxWell Biosystems and we are innovating the next generation electrophysiology platforms and solutions. And since more than 20 years ago, the technology has already been developed and matured in Andreas Hürlemann's lab at ETH Zurich, and we have a good publication record before starting the company in 2016, when we thought now it's ready for everyone to use it. And since then, we have in-house expertise on hardware, software, and also applications of these electrophysiological platforms. And our office, our headquarters is here in Zurich, so we are broadcasting live here from Zurich, and we have our offices and labs as what you can see in the video. And we have users around the world, academic labs, biotech, pharmaceutical companies, and also contract research organizations. So for today, I would like to let you know that we have every year since 2021 our in vitro 2D and 3D neural network summit. And this year we had it fully on site. So you can see the video here. And we have also our sponsors. Thank you so much for all of those who joined us during this event. And you can see that we have talks from users and also non-users, everyone who would like to be part of the neuronal networks community. And also we have some poster sessions as well. So please, if you want to join us next year, save the date, April 8th to 10th, 2024. It will be here again in Zurich, Switzerland. So you're all welcome to come and join us. Okay, so one of the speakers in our summit will be also our guest speaker for today. And I would like to have the outline for today's webinar. So first, I will introduce to you about high-density microelectrode arrays, this technology, and then we will go on to our presentation, which will be about 2D human stem cell-derived neurons. So you will know more about it soon. Afterwards, we will have our Q&A. For all of you who are new to this platform, so you will see right below there are some icons. So there is, of course, a way to chat. I see that some people already started chatting. So you can go on and just say hi to everyone or just mention your comments, some discussions. But then if you would like to have some questions answered at the end of the webinar, please add them inside this Q&A. So there is a Q&A section. Just add them there and we will go through them in the end. If in case we are running out of time, we will be able to answer these questions by email and send you directly. So please let us know who you are so that we can send these answers directly to you. But we will try our best to answer all of the questions in this webinar today. All right, so I know you're excited. I'd like to first introduce our speaker for today. We have Dr. Rouhollah Habibey from the Volker Busskamp Lab in University of Bonn in Germany. So I'd like to just stop sharing and say hello to Rouhollah. Hi, Rouhollah.
Hi, Marie. Hi, everybody.
Yes, so he will be back later on and he will have the presentation together with us live. Okay, so let me share my screen again. So today, Rouhollah will talk about long-term morphological and functional dynamics of human stem cell-derived neural networks. So this is really a fantastic work, which he also recently published. So you will see the paper later on getting added in the chat. Okay, so now let me proceed with the introduction to high-density microelectrode arrays. So here at MaxWell Biosystems, our motto is every cell has a story to tell. And there are two main technologies that are being used to be able to characterize cells, especially neurons. On the left side, we have imaging, which allows us to see the cells, identify the different cell types, see how they grow, and how to connect with each other. On the other side, we have patch clamp as the gold standard for electrophysiology. So we're really able to get high quality signals from each individual neurons that you're able to get close to with a patch pipette. Now, our technology here at MaxWell is high density microelectrode arrays that allow us to be able to also generate these images, just all electrical based on the activity of the neuron. And from here, you can see it's an action potential initiating here and propagating along all of the axons. Plus, you can also get detailed information such as the spiking information, amplitude, firing rate from every neuron that you're recording. So really, it's very complementary to these two technologies and something that goes in between. So you can also use it for all of your neuron experiments. All right. So for all of you who are familiar with microelectrode arrays, the classical standard ones are these low density MEAs, wherein sometimes you miss some neurons, or sometimes you're not able to identify whether you're recording from one neuron or multiple neurons or like 10 neurons at the same time. Also, there might be a chance wherein there are some electrodes that do not get any signals at all. While for high density microelectrode arrays of MaxWell Biosystems, we have many electrodes tightly spaced together. It allows us to be able to identify the signals for every neuron that is on top of the array. So here you can really see the signals coming from these yellow, blue, green, and red neurons individually. Plus, because we have these small electrodes, you can also follow these propagating signals across the axons. So just to see it in terms of microscopy, so here at the back, we have the electrodes, and here you see at the higher Z stack the neurons. So every neuron can be recorded by multiple electrodes at the same time. So this is really the power of high resolution from MaxWell Biosystems. Now, what are the key advantages of using these many electrodes? So first, you're able to get accurate activity maps. So not only just having information of yes or no, but you really have more detailed information. So you're almost doing some sort of imaging without doing any labels on your samples. So here you can identify the location of the largest signal, which are then you can see okay, there are 4 neurons, and you can see it right here. Plus, you can also get more detailed population network bursting profiles. So if you have less electrodes, maybe this is not as pronounced, but if you have access to every neuron, you'll have more better looking profiles for your samples. And finally, which is the most exciting for us, because it's a novel readout, is to be able to identify the subcellular conduction of action potential along the axons. And it's really made possible because of the tightly spaced electrodes that are having very sensitive, high sensitivity to the signals. So in the end, we can really see all of the signals, all of the cells, identify the activity of each cell, and really have access to these small signals and be able to read out at different scales from network to cellular level down to subcellular level. All right, so how does this look like in terms of product? So we have two products at MaxWell Biosystems. We have the single well, which is called the Max1. It's a very flexible platform. You can use it for cultures and special applications. You can put it inside of your incubator, and it's very small. It's like the size of your phone. And you can also attach a perfusion system to be able to do some acute recordings from brain slices or other samples. So you can see here below the different chips that we have that is compatible with the Max1. On the right side, we have the Max2 system, which is the multi-well version of our Max1 system, which allows simultaneous long-term studies for cell cultures, organoids, etc. And the advantage here is that we have already a built-in incubator in this main frame. The temperature is controlled, and there is a CO2 inlet. And this mainframe is compatible with both 6 and 24 well plates. So you can really do parallel experiments, which is very similar results as what you can get from the Max1. So how is that? That is because every well has the same chip. So in every well, there is no difference whether that is in Max1 or in the Max2. Every well has 26,400 electrodes per well, and that's within the 2 by 4 millimeter square area. And the distance between these two electrodes is 17.5 micrometers, which really allow us to access these small signals from every neuron, as you can see here. Plus, what's really important is that even though we have these small electrodes, we have low noise readouts, which means that we have high sensitivity to signals, even though they are small and propagating. Additionally, and also especially, any of these electrodes can be used for electrical stimulation, which means that you can target specific neurons to be able to evoke them and see how they are responding to the stimulation. All right, of course, not just the hardware, but also the software is important for these microelectrode arrays. So we have a software which is called MaxWell Live. And here you can see the 26,400 electrodes. Whenever you see a red triangle, that means an action potential is detected in that specific area. And you see sometimes all of the cells are getting active, and sometimes individual ones. And you can really zoom in to the activity of every spot. So here you can see these cells are active. You can see there's a burst sometimes. You can even zoom into the activity of an individual neuron, such as this one. So you can really ensure that the signal that you're recording is biological and not just noise. So you can also use the software for organizing and scheduling your experiments, and also do some analysis and generate summaries. All right. So these are the different assays that you can do with our software. So activity scan, network assay, and action tracking assay, all automatic within the software. So if you would like to learn more, just contact us. And the system has been used and proven in many high impact publications. So as of today, we found 158 articles, but we know there are more. And these are some examples of them, high impact papers on 2D cultures, organoids, etc. And in conclusion, both our systems MaxWell and MaxW can be used to characterize neural networks at the network, cellular, and subcellular levels for any of these research fields or even beyond. All right, so now I'd like to transfer the floor to our guest speaker, Rouhollah Habibey, who will talk to us about his work on long-term development of human IBC-derived neuronal networks. Okay, Rouhollah, hi.
Hi, everybody. Thank you for inviting me. And also, I would like to welcome everybody who is attending this webinar session. So if I go for sharing the...
Perfect. Yes.
Yeah, that works?
Yes.
Good. So today I will talk about long-term characterization of human iPS-derived neural networks. It is both morphological and electrophysiological characterization. But before going to my talk, for inviting me, and also I would like to welcome everybody who is attending this webinar session. So if I go for sharing the...
Perfect. Yes.
Yeah, that works?
Yes.
Good. So today I will talk about long-term characterization of human iPS-derived neural networks. It is both morphological and electrophysiological characterization. But before going to my talk, I also would like to introduce briefly our lab. I am in the Professor Busskamp's lab at University Clinic Bonn in Germany. So the main theme of the research in our group is engineering different types of neural cells based on transcription factors or expression. And we have highly focused on retinal cells, especially on photoreceptors, directly from stem cells, or we are also growing them inside organoids to achieve a higher or mature phenotype. In our group, I'm mainly involved in engineering neural circuits. We have this nice toolbox of neural cells. We combine it with different type of microfluidic devices to structure network morphology and mainly characterization of functional properties of this engineered neurons or the circuits that have been derived from these neural cell types. We were mainly using standard MEAs and high-density MEAs for this purpose, but we also have a single-cell characterization like Busskamp. We also have a handful of optogenetic tools for full-field stimulation or controlling the whole network activity. And we, in the same time, we have this holographic optogenetic stimulation system that gives us possibility to spot individual neurons in the context of the network, which is something that I will briefly talk in the first part of my presentation today, and then I will go for our long-term data on high-resolution MEAs. So the main neural or neural cell types that we engineer in our group is glutamatergic INGN-derived neurons. These are based on PGP1 cells, and they are overexpressing NeuroG1 and NeuroG2 under Tehran promoter. And by adding doxycycline to the culture media, we can pre-differentiate the cells in less than 4 days in vitro. The cells have already been characterized in the single cell level using Busskamp system, which if you are interested, you can find the data in this paper that I put here from Rebecca Lam. So how I use this stem cell-derived neurons for our long-term cultures on the standard MEAs. First, we seed the cells on matrigel-coated plates. We differentiate them by doxycycline, and then we remove undifferentiated cells using RSC. In parallel, we prepare astrocyte in a separate well or flask, which are first in the astrocyte media, and then we change the media to brain phys medium that which is also common medium for the differentiated neurons, which I will discuss later. But I also want to mention here that astrocytes that we are using, or I am presenting here, they are rat primary astrocytes. So after 4 or 5 days that the neurons are differentiated, we detach the neurons and we bring them to the PDL-coated MEA substrate. The reason is that the matrigel coating is not stable for long term. So if you want to perform long-term experiments like tracking the network activity development or applying optogenetic stimulation at different time points, we need to have this kind of stable coating. After reseeding neurons, as you see here with red cells, we flip the astrocyte culture on top of the neural culture. We use this kind of paraffin dots, which is called banker culture system, the whole system. And therefore, the neurons are already exposed to the astrocyte secreted factors, which are supporting neural maturation and improved functional properties. In case you want to do optogenetic stimulation in the networks before induction, we transfect the cells with, for example, in this case, E5P tag rhodopsin based on lentiviral construct. So after adding the lentiviral construct and expressing rhodopsin, as you see here, this is before reseeding. Then we detach the cells and we bring them onto the MEA substrate, and after a while, it is possible to perform optogenetic stimulation. For example, here you see the traces of the activity in response to 50 millisecond applied pulse, which has been applied at different frequencies. For performing simultaneous optogenetic stimulation and electrophysiology recording, the whole MEA amplifier and chip is mounted on the inverted microscopy stage, in which we can apply full field stimulation from the bottom, and the holographic stimulation has been mounted in an upright manner by Felix Schmieder. He's a former PhD student, currently is postdoc in Professor Cesar's lab. So holographic stimulation uses this liquid crystal spatial light modulator. In general, the light is collimated through this first lens onto the beam splitter and projected onto the spatial light modulator, and then it is focused through the telescopic lens onto the sample. I would like to mention here that because we needed to spot individual neurons, we prepared magnified microscopy images in another microscopy, and then we, after saving the coordinates of the individual neurons in a method-based GUI, we focused on the electrode level to ignite or evoke individual cells in the context of the network. It is possible to do simultaneous full field and holographic optogenetic stimulation, but we were mainly recording the baseline recordings and then applying full field stimulation, and then we were going for the holographic optogenetics. So as I mentioned, we have this long-term platform that we can culture cells for more than 90 days, record at different time points, or do microscope imaging, and we applied full field stimulation and holographic stimulation at 5 different time points for the collection of 4 samples. And here you see the full field stimulation that evoke the whole network in the same time, but for holographic stimulation, we chose this spots beforehand. So before going to the holographic stimulation, I briefly explain some data about full field stimulation. The blue marks here, you see that there are the light pulses that apply to the network, and these are the data raster plot of 10 selected neurons. As you see here, we have the coupled responses during the light pulse, and there is also the tail of the activity coming after light pulse in the light off period, which is related to mainly to the multisynaptic activation. So what we found in this full field simulation, which is also natural, that the number of detected units or extracted neural units are higher in the presence of full field stimulation, which is a good point because it means that it is possible that we can use optical stimulation for improving the network functional maturation. If we engage further neurons in the pool of the spontaneous activity or evoked activity, then it will improve the network functional properties. And another obvious data that by full field simulation, we have higher level of activity compared to the spontaneous condition. And as I mentioned before, if this is an exemplar post-stimulus time histogram, the blue one is representing the baseline without blocking the excitatory synapses. But in presence of the excitatory synapses, we see that the tail of the response to the optical stimulation is disappear, and only we see the response in the 51st millisecond or during the light pulse. So before applying the holographic stimulation, as I mentioned, we prepare this large image from the network, this collection of 20 to 30 mosaic images. Then we identify individual neurons to save their coordination in our custom-made software. And then by running the software, it was applying 20 to 30 pulses for each neuron. Let's say the first neuron is receiving first 20 to 30 pulses, then there was an off period, and then the system automatically was going to the second neuron, and so on, until all spotted neurons in the context of the network has been stimulated. This graph represents the response in one of the selected electrodes to 550 applied episodes or 550 stimulated neurons. And as you see, only in few of them we observe the response to the holographic stimulation. And the same for the extracted neurons. Not all neurons are responding to all holographic episodes, only the ones that we targeted shows the response to the holographic stimulation. And another point that I want to mention here is that not all holographic stimulation episodes were successful, and it comes from the fact that even though it is possible to stimulate the neuron with most probability, it is difficult to get the activity or evoked response in the electrodes because the resolution of the standard MEA is lower. We have only 60 electrodes to read out the activity. So we also compared the activity between holographic and optogenetic stimulation, and as we expected, we observed a lower response to the holographic stimulation. But here I put a descriptive explanation of differences in the network activity or the data that we can extract based on holographic stimulation. So first, the full field stimulation, if we look to the post-stimulus time histogram of 3 selected electrodes, we see that all electrodes are showing responses right in the applied stimulus, and then there is this tail of the activity afterward, and it comes from the fact that all channel rhodopsin expressing neurons are responding to the light pulse. So the whole network is stimulated at the same time. But if we apply holographic optogenetic to the neuron that is close to the electrode 24, we see more or less similar profile in this electrode with the direct response and indirect response coming in the light off period. But this direct response in 2 other electrodes has been disappeared. We can see some indirect response which is indirectly connected to the neurons in the another electrode. And if we change the neuron and stimulate the neuron close to electrode 45, we see this tiny direct response appears in the electrode 45, which is not the case in the previous stimulation. And the direct response of the electrode 24 is already disappeared and only shows the indirect response. So based on this different episodes, it's possible to see how each of these individual neurons is connected to which one of the recording electrodes. Therefore, if I summarize it in this image, we apply the stimulus to a neuron that is far from the recording electrode. Therefore, it is not possible to capture its activity, but it is indirectly connected to the neuron that is this electrode is recording from. Therefore, by light stimulation, we can observe indirect response in this electrode. So we try to do this kind of analysis for all applied episodes. For each episode, we extracted this kind of connectivity traces. And by pooling all these episodes of holographic stimulation, it was possible to extract kind of network connectivity map. As I mentioned before, and you say that blue lines are representing the neurons that we were able to find a significant or meaningful connection between those neurons and at least one of the recording electrodes, and the white arrows are representing the examples of the neurons that were stimulated, but we couldn't find the connectivity or activity in the corresponding electrodes. So we tried this at different time points, and as you see, over time, there is a large differences in functional connectivity maps over time. This can be related to 2 points. One point is that the activity itself takes different trajectories. It has a lot of fluctuations over time, so it can affect the network functional connectivity maps. But the other thing that I was more interested and I want to highlight here, which is the rest of the talk also is about that, is the structural changes in the network that is happening over time. If as I mentioned before, we did the microscopy images over time from these networks. And by assembling the microscopy images on top of each other, we were able to see this changes in the network morphology over time, which causes to the fact that neurons or the cluster of neurons can move from one electrode, therefore their activity can disappear in another day of recording, or the new activity can appear in the electrodes that were silent in the days before. If we focus in this area, this is the magnified view of the area in the right side, you will see that the neurons are coming toward this electrode. These neurons will move toward this recording electrode, and neurons that are beside this electrode will move far from the electrode. Over the course, these are the data over the course of 2 months. So what this causes, it causes the problems that if we want to have concatenate the activity over long-term period, it is really difficult to extract this stable or reproducible data from them. So that was the reason why we were interested in high-density MEAs with high resolution, because it was possible to track the activity over time in these networks. But because before going to that, I also want to show you 2 examples of our previous data. When I was in Italian Institute of Technology, we had this system that was able to record the network morphological changes in a continuous manner. This is an incubator-independent perfusion system, and it is hippocampal neurons for 14 days continuous recording of the network morphology. You will see how network structure is shifting or changing by culture age over time. And recently, I tried this inside microfluidic devices. If you focus on this part, you will see the network will cluster in that part, which causes the neurons to shift their positions, go far from the electrodes, or displace on the surface of the sensor. So we try to adapt the long-term culture system to the high-density MEAs. The protocol was more or less similar. The only difference is that we keep the astrocytes in separate environment, but every week we were supporting the cells from by astrocyte conditioned media. For microscopy, as you already know, we have not the CMOS system is not transparent, so we need to do upright microscopy. For this, we had this transparent PDMS lead, which had very thin sealing, for it was possible to do upright microscopy, because normally to perform microscopy from the whole sensor area, we had to do mosaic images to have a better high resolution images from the whole sensor area. So we need to spend a little bit more time on the microscopy stage. For that reason, to prevent from evaporation, we had also to better sealing of the environment. So this assembled images at different days were used for tracking the network morphological changes over time, which is more or less like 2 months tracking the network morphology, and in the same time we were performing activity scan from the networks. We were performing it in full scan mode to extract the network. Later, this data were used for electrophysiological analysis, but at the moment I want to focus on network activity scan and how it is affected by morphological changes in the network structure. The bright areas that you see here, they are the areas that are with higher fidelity, represents the areas that the cell bodies or cluster of the neurons are located. So by this, we can track also the effect of structure on the function over time. This image represents the network activity at day 22. Neurons are nicely distributed, and you will find out later that how these neurons are coming together to form smaller clusters after 1 month, and then these smaller clusters joining together to make bigger clusters. And actually, the shifts in the position of the neural cells are coming from this pull and push forces between the axons. For example, in this part, you saw that the cells are joining together, and the smaller clusters are merging to the bigger clusters. We track the network functional property or functional image over time in the same MEA, and you will see exactly the same movement happening over time, which shifts the position of the neurons. So we see that how network functional image can be affected by network morphology, and this is the overlaid image from microscopy and also functional image. You see how with higher fidelity, as the clusters of the networks are moving across the network, across the chip, we also see this shift in the place of the functional map. Each of these yellow squares are something around 100 micrometer, which is around 7 electrodes of the MaxWell chip. So by looking to this data, we can say that one of these clusters, for example, shifted something around 100 micrometer on the surface of the sensor. It means that over time, different set of electrodes are recording from the same cluster, and activity in the electrodes is changing. So by we have the different frame of the network morphology over time, it was possible to locate individual neurons and track their activity trajectories, and based on this based their morphology or movement trajectories, and based on this trajectory, it was possible to calculate the displacement of the neurons on the surface of the sensor. This is for 2-month period of recording. Here also you observe the clusterization of the neural cells on the surface of the substrate. So we have this data. Based on this extracted data, it is possible to measure the displacement, which in the earlier days of the network development, it is quite higher, but later For example, around after 2 months post-induction, we see that this displacement is decreased, and it is always after formation of the larger clusters that are more or less stable. And this is the cumulative displacement for the percentage of neurons that showed specific displacement for the course of 2 months. This is more or less something around 100 to 150 micrometer in more than 70% of the neurons. And I should also mention that the data that you see in the upper panel is combined from 3 MEAs, but the displacement of the neurons or the clusters are not same in between MEAs, which could be somehow related to the coating. So coming back to the electrophysiology analysis, here you see the exemplar profile of the activity at different time points, which appears as individual action potentials, then develops to this longer bursts, and then burst activity. And if we look to the activity in the extracted neural units, at the same days post-induction, in each day we have each row of this, each dot is one action potential, and each row is representing activity in one neuron. And in total, in each day we have 10 neurons, the data of 10 neurons aligned. We observe that the local bursts are appearing around after first month post-induction. Then the synchronized long bursts are appearing in the second month, and in the third month, the synchronized activities are becoming quite packed with higher frequencies. I should also mention here that based on the morphological data, this is the time points that we have formation of the smaller clusters, and later on the older cultures we have the larger or bigger clusters formed, and it could be somehow relevant to the network structural changes over time. And this slide summarizes the activity profile of the network over time. I want to highlight this point that in all cases, network activity increases up to day 50 to 60. There is a peak at day 50 to 60, which also shows higher burst duration at that moment. And then the action potential frequency declines a little bit, but remains stable for the rest of the culture age. And this is also a similar phenotype actually we observe in the primary networks, for example, in rat cortical or hippocampal neurons. Around day 20 to 25 in vitro, we see this peak of the activity, and then the activity declines a little bit and remains or fluctuates around this stable line. Regarding the burst frequency or burst duration, we see that the burst frequency is increasing, as I showed also in the raster plots, and the duration of the burst increases or take a peak, and then it declines over time. This is kind of compensation for the increase in the burst frequency. So I will summarize the data here. Our holographic optogenetic system shows superior resolution compared to the full field stimulation. It can be used nicely for extracting fine or smaller network connectivity motifs, but standard MEAs were not compatible with this high resolution stimulation. So we will try to apply this system for higher resolution readout like CMOS systems. Another point that I didn't mention during talk, the main idea of holographic stimulation is to have access to stimulate several neurons in the same time, or these neurons are selected already, so we can try to apply stimulation to, for example, 2 or 3 or even more neurons in the context of the network to see how they are connected to each other. And to have a more versatile system, it is also possible to use different optogenes and apply or play a little bit with wavelength of the light. So it is possible also, for example, we can apply inhibitory optogenes to block the activity in some neurons and induce activity in another neurons to extract this fine network connectivity motifs. Our long-term data on the CMOS MEAs showed this large-scale changes on the developing network structure, which was also in the same time in parallel to shifts of the neural cell position on the surface of the sensor and coming from the push and pull forces between neurons. As neurons are becoming clustered, it applies these forces on the individual neurons. So it is important to see the effect of this long-term morphological changes on the functional readout, especially if we want to concatenate signals of the same electrode over time to extract, for example, units or individual neurons from the same neuron. With mass probability, it should not be the case because neurons are moving on the surface of the substrate, and by simple concatenation, maybe it leads to the problem in the collection of the data, because definitely one electrode is not necessarily recording from the same set of the neurons over time. One thing that I mentioned, we always were keeping the neurons in a separate environment, or even though they were in the same environment with astrocytes, we were not mixing them together. In the past, it was for collection of the molecular biology data from pure neural populations. But in the ongoing experiments, we have integrated astrocytes in the neural cell models to see that if they are affecting or they can help to keep the network structure stable over time. With this, I want to thank our previous lab members in Technical University of Dresden and current lab member at University Clinic Bonn, especially Johannes, who helped me on the second part of the project, the data that I presented here today, and Professor Cesar's lab for assembling and helping us in the holographic simulation system, and funding agencies that supported my position, and as well the setups that we are using, or the data that we collected by these setups. And thank you also for your attention. I am happy to take questions.
Thank you so much, Rouhollah. Really, really amazing talk and fantastic, fantastic data sets. So as like in our morning session, we actually have a lot of questions popping up right now. So let me go through one on one and then so before that, I would like to ask the audience to please answer a few questions from us through our poll. So I think we can now start up the poll. So first, what is your current main application? Are you working with 2D cell cultures like Rouhollah, or 3D cell cultures also like Rouhollah, tissue explants, in vivo, or others? So we would be very curious to hear about what you're currently working on. All right, thank you so much. So next question. Are you currently using MEAs, microelectrode arrays? Thank you. Thank you so much for answering. And then finally, our last question. We have field application scientists here at MaxWell Biosystems. Would you be interested in getting a tailored presentation or demo of the system if you don't have one yet? Yes, I know that in our audience, there's already a lot of users. So thank you so much, everyone. All right, so now we can start answering the questions. So once again, please add your questions in the Q&A section so that we can see them and answer them together with Rouhollah. So first, let me start from the question from the morning session, Rouhollah. So there's one remaining. There were too many questions, so we are answering them now. So regarding bursting activity of neurons on the MAX1, did you see any prominent bursting, especially with the astrocyte conditioned media? I guess this is in comparison to without the astrocyte conditioned media.
Normally, the burst activity As I showed in the raster plot, it appears around day 30 to 40. Let's say like in general manner, like local bursts, and then we have this highly synchronized bursts. I had like this data that I presented here today, I think it was around day 90, but these cultures, we also kept them for longer time. They were normally showing burst activity as long as the cultures are live. Yeah.
So do you think they are earlier because of the astrocyte conditioned media, or without the astrocytes, the burst would come much later?
We have not tried them without astrocyte conditioned media yet. We always were supporting them with astrocyte conditioned media, but currently we are trying them with mixture of astrocytes. It is giving better results compared to the conditioned media. Yeah.
All right, thanks a lot. So this is more of a technical question that is being asked by Tammy. Hello, Tammy. So thank you so much for all of your comments. Ashley, I think Tammy also said this presentation is amazing. Thank you, Raula. So
Thank you.
yeah, so she's asking about the copy of the presentation and the video. So I think this will be sent by email later on for the session that you have attended. So please wait for that. Okay, now next question is from Richard. Please describe again the optogenetic system. Maybe you can share your screen and then show the holographic setup. Thank you.
We have the full field stimulation, which is like in an inverted manner. For optogenetic stimulation, it is the light source is the 450 nanometer laser diode. I mean, this about very technical details of the setup, I can't say exactly how it looks like, but because the system is assembled by our collaborators, just I don't know what exactly the question is regarding this device. I should say that here we were performing it in an upright manner, and the distance between the objective and the sample was quite higher. So there was something around something around 1 centimeter distance between objective and the sample, or the neural cell plane. Extra things that I can explain about the holographic stimulation, except this specific pattern of the generated light is focused through the lenses onto the sample. And if I go, I think maybe I explain it better in this slide.
Yes, yes.
So what we were doing, we had this the holographic optogenetic system already has been automatized by MATLAB-based UI. So we were just loading the coordinates of the selected neurons to the holographic stimulation system. After selecting all neurons, the image has been prepared in another microscope. The coordinates of the neurons were already saved, and then we were aligning this image with the live image of the holographic stimulation microscope. Then the locations were loaded to the microscope, but before performing this stimulation, as I showed you in the previous slide, the plane for selection of the light focus on the sample, we were trying to only focus on the electrode, on the visualized electrode level, to match this Z plane together between the already taken image and the live image on the microscope. The rest of the steps were quite automatic.
Okay.
Yeah. I don't know how much it's helpful, but about very technical details of the holographic stimulation, I can't say more than this.
Yes, and I think you have some references, so maybe we can also just send later.
Exactly.
Thanks a lot, Raula. Next, we'll move on to the next question by Aldana Gojanovic. So how many cells did you plate on the chip?
Normally,
answer on the max one.
I don't recall exactly the number of cells that we plated on the MaxWell chips. Normally, we see them something between 10 to 20,000 recently, but in the past we were seeing them with higher densities. And it also depends on if we want to add the cells only into the sensor area or into the bigger surface of the MEA. In the data that I presented here today, it was larger area of the sensor, and I think it is around 50,000 neurons at least. But it should be in the paper. At the moment, I can't recall it, the exact number of the neurons. Yeah.
Thanks a lot. Okay, next we have a message from Thanasis Alexandris. So thank you so much for your great presentation, Raula. To what extent do you think changes in the network activity are due to the changes in the neuronal clustering versus the maturation of the synaptic formation? It seems there is a very high correlation with clustering, like the bursts appear because the clusters appear.
Yeah, actually, we were like we didn't know what will happen, so we just wanted to check the. When I started this project, I was mainly interested to see how neurons are shifting on the surface of the substrate. And we observed this cluster formation. And then we were compared our data for already published data that I don't recall the name of the paper. Maybe later I can write it here. There was this group that they have tried to compare the nicely distributed networks with the networks that engineered to be clusters or
Is it
formed
also a
modular.
German?
Yeah. No, actually, they didn't use microfluidic devices. I also, in our microfluidic chips also, we try to have this kind of modular structure in which neurons are clustered here and there, and then they connect through the axonal bundles. And we observed that this clusterization actually leads to increased network functional properties. Now the question is that about the random networks, if the type of the functional connectivity pushes neurons together, or it is the activity, the morphology that enhances the activity profile. I think if we go to the brain development, in the brain also, based on this economic of the network development in the brain circuits, neurons that are functionally connected together, they are tended to come toward each other. And actually, there is this theory that folding in the cortex is also coming from this push and pull forces of the axonal bundles, and also neurons that are functionally, let's say, coupled together to perform the same task at the end.
Okay. Thank you so much, Rala, for the answer. Next question. Sorry, we still have quite so many. So this is from Tammy Soliman. So were the LED and laser stimuli responses distinct due to different types of neurons? So did you target different cell types?
Yeah, sorry, I didn't get the question.
So in your light stimulation, so you had both the full field LED and also the laser holographic stimulation. Were they kind of targeting different types of neurons?
No, I would say no, because we used LED illumination for full field stimulation. So it was normally all neurons that were expressing channel rhodopsin should have responded to that unless we apply very high frequency signals, so the channel rhodopsin is cutting saturated. But about holographic stimulation, we used a laser light source, and for every episode of stimulation, we were just spotting one neuron. Maybe I forgot to mention that the width of the light beam has already been calculated, and it was around 8 micrometer. So
very small.
Yeah, exactly. It is possible to only apply stimulation to individual neuron without inducing the activity in the surrounding neurons in the network. That was the idea of the holographic stimulation.
All right. And all of the cells that are seeded, that are neurons, are all expressing channel rhodopsin.
I would say no. We also measured that, by average, something around 50% of the neurons are expressing. We were using lentiviral construct, which was giving quite good yield of channel adoption expression. The microscopy images are also, that's the reason I didn't present here also the bright field images. We also were preparing bright field images. And another point that maybe it's worth to mention here is that the expression level also is not same in different neurons. Some neurons have higher level of expression compared to the others. And these are the facts that is difficult to make it more homogeneous, I would say, to express in all neurons or higher percentage of neurons. These are all technical challenges, or in the same level in all neurons. This is always challenging, yeah.
Thank you. Okay, we're going through a bit more questions, Rouhollah. So this is from Hyap-Chun Hao. So do you observe any phototoxicity effect when you optically stimulate the cells, both in the control and the optogenetics line? So is there any increase in the laser intensity? And if you do so, is there increased firing in the burst rate, or even if you do that in control without the channel rhodopsin?
Actually, the data that I put from one electrode, if we were always checking, because holographic stimulation with 400, 500 episodes of stimulation was always taking quite long term. It was also interesting for us to see that if keeping network MEA outside in ambient conditions can affect the network activity. So that's the reason we also track the activity over the course of whole episodes. So so far we didn't see large changes in the network activity or electrode activity over time. And I would say that phototoxicity in the case of optogenetic stimulation should not be the case, because the light is not distributed across the network. It was only inducing individual neurons. Maybe it is the case for full-field optogenetic stimulation, but in our case, we were only applying less than 3 minutes. Yeah, optogenetic stimulation with quite long time points. It was something around 0.5 hertz that we applied. So far, we didn't see. I mean, if we would have seen the phototoxicity, we should have seen also a decrease in the activity over time in our networks, which was not the case.
Yes, like some cells would not respond anymore, or breaking connectivity across the different cells. All right, really great results. Thank you. We have another question from Elisa Tentori. So can you describe again the functional connectivity reconstruction with your holographic stimulation, the one with the graphs? And thank you for the amazing presentation, according to Elisa.
So what we were doing, to find out the first step was to find out if we stimulate the neuron, the response in the electrode is significant. So what we were doing, based on comparing the baseline activity, because we were using the spaces between applied light pulses mainly to measure the baseline activity. If during the light pulse we had more than 6 standard deviation increase in the activity level, we were considering it as a meaningful response to the optical stimulation. So for full field stimulation, we simply used cross-correlation analysis to find the correlation between electrons that I didn't present here, but it is in the paper. But for holographic stimulation, if there was a meaningful connection between a stimulated neuron and a recording electrode, then based on the strength of the connection and the presence of the connection, it was plotted. So it was more or less based on the increase in the activity level, and if this activity is significant compared to the baseline. So it was performing for each individual episode, and then the whole episodes of the stimulation were assembled together.
Thanks a lot. Yeah. Okay. And this you also did spike sorting for sure, not just per electrode. Yeah. All right. I'll just go through all the questions as much as possible. I know we are running out of time. So next, from an anonymous attendee, did you see any responses to APV alone versus NBQX APV co-application? And in what day in vitro did you see the APV alone response in the induced neurons?
Unfortunately, we didn't write them separately. We were always mixing them together because we were more interested to block the whole excitatory synapses.
Mm-hmm.
The cells supposed to be glutamatergic neurons.
Yep.
It has been proved. So, and the main idea was to see that if in presence of optogenetic stimulation, we still have this tail of the activity, which or to be sure that it's coming from the multisynaptic pathways. But we did it. It's a good idea to check if which one of the receptors are expressed or if both of them are expressed, but we have not tried it separately. Yeah.
All right. We have one question from Roy Ben Shalom. Hello, Roy. He said, thank you, really impressive and insightful data. So this is in connection with the functional connectivity before. Can you apply it also on the HDMEA data? Or have you tried it already?
Functional, yes, definitely. I think there are other groups that have tried, even on organoids, they have tried to extract the functional connectivity maps. The principle is the same. It is based on cross-correlation analysis for spontaneous data, but for optical stimulation, we use different type of analysis, as I mentioned before, based on the significance of the connection between electrode and the neuron, stimulated neuron. But it should be visible on the CMOS MEAs also.
Okay, and once you try also the holographic stimulation on the high density MEAs, I hope you can also see relevant results. All right, so that's it for our Q&A. I'd like to also tell everyone that we are celebrating our 7th anniversary at Muscle Biosystems now in September, so please wait for our promotions. And thank you very much, everyone, for joining our webinar today. Thank you so much, Rouhollah. Really, really amazing talk, and thank you for answering all of the questions. It is our pleasure that you are part of this webinar today. And for everyone, please contact us at info@mxwbio.com if you have any questions. Also, if you have any questions directed to Rouhollah, either through us or directly contact him, please go ahead. We have the paper also in the chat. You can also schedule a call directly with us at Calendly or visit our website at www.mxwbio.com. And you can meet us at any of these upcoming events. We are still filling it up. So if you have any events that you also want us to join in, please let us know. All right. Thank you, everyone. Good morning in the US, and have a nice afternoon later, and have a nice evening for all of us here in Europe. And if someone from Asia is still here, have a good evening and good night. All right. Thank you, everyone. Bye.
Bye.